Yes, I'm going to talk about transgenral epigenetics, and it's actually part of a discipline called genetics. So you can always rest assured there is nothing magical in here, except that, of course, genetics is a bit more complicated than just studying variations in DNA sequence. And because Christophe asked me to give a bit of an introduction, I have to go through a few slides where you all realize that what we have is not just DNA sequence in our cells, it's what we call chromatin. And of course, this is a big issue in eukaryotes, not so much in bacteria, because bacteria more or less do have naked DNA in their cells, not completely, but almost. And chromatin is not just a packing device, it's indeed a very complex structure which is quite dynamic, made of DNA and histones in particular, and a lot of accessory proteins that bind to this nucleosome unit of chromatin. And both DNA and nucleosomes can be subject to a whole variety of modifications, and this modification somehow is what makes chromatin more accessible for transcription or less accessible for expression. So that's where you have genes turn on or genes turn off in relation to chromatin states. And here, it's, there is no point in going through the details here, but suffice to say that here you have the nucleosome structure, which is a basic unit of chromatin, with the DNA wrapped up around a core of 8 histone proteins. And you have modifications on the DNA, or you can have modification on the DNA, which is the addition of a methyl group on cytosine. So you get this 5-MC, or 5-methylcytosine, which usually referred to as DNA methylation. And then on the histone tails, often, what you can have also is a series of modifications which go from ubiquitination, methylation, phosphorylation, so on and so forth. And in addition, even though that histones are extremely conserved among eukaryotes because they are the basic units of chromatin, you see a lot, you can find a lot of what we call histone variants, and which again bring a lot of flexibility to chromatin and make it more permissive or less permissive for transcription in particular. And it's not just transcription, which is regulated by chromatin, it's of course DNA replication and all the activities that take place on the DNA molecule in a eukaryotic cell. And a very naive cartoon, what you can consider is closed chromatin and open chromatin. Now, this is what makes a given genome giving rise to multiple epigenomes. And in a way, this is an answer to the question or to the program set up by Waddington about epigenetics. How does a gene bring about a change in phenotype? And it brings about a change in phenotype not just like that, by changing its DNA sequence, but by changing the chromatin states associated with the gene sequence, and that's how you get cell differentiation in particular. So for one genome, we have, of course, multiple epigenomes, but even this notion is actually not completely true, because we know that our genome is not constant. There is always DNA sequence mutation that occur at every cycle of replication. So not everything is fixed, even at the DNA sequence level. Now, where does this definition of Waddington sort of shift towards this Holiday's definition that Francesca referred to, is this notion of memory. And indeed, what we know is that chromatin does convey memory to a cell, and not only to the cell, but to its progeny, and through multiple cell divisions. And a beautiful example can be found in plants, where you have this process called vernalization. You know that wheat, what we call winter wheat, for instance, has to go through the winter in order for it to flower at the next spring. And this process, at least, has been worked out in great detail in another plant species called Arabidopsis. And what was found is that you start with one key gene which controls flowering, which actually represses flowering. And this gene, of course, has to be turned off early on during development, because you don't want to flower as soon as you germinate. So this gene sits on a piece of chromatin, which is extremely compact, close, early during, soon after you germinate. And somehow, I got it wrong. This gene actually starts open for whatever reasons, but through the cold gets progressively shut down by getting the chromatin more and more compact. So this occurs through the winter over a prolonged period of time. And now when the spring comes back, even though you get back to hot temperature, somehow this chromatin state now stays compact through multiple cell divisions. Now you have the plant growing very fast, and now you have memorized these compact chromatin states. And actually the activities associated with this compaction or with the memorization of these compactions are called Pierce polycomb type repressive complexes. And I think Giacomo this afternoon will tell you a very nice story about polycomb repression, which typically is reset at every generation. At every generation, even though you end up with a plant that has this very compact chromatin which allows flowering, at the next generation you will start again with an open chromatin. So at every generation, you compact your chromatin through the cold and you reopen it through sexual reproduction. So that's a key element of reprogramming. Now we know that reprogramming is indeed extremely important, and the best example is in mammals, where we have now DNA methylation associated with a whole range of gene regulatory processes. And what we know is that methylation is not constant throughout development. Indeed, it starts very high, both in the sperm and in the oocyte, but as soon as you fertilize the egg, now you see that there is a huge drop in DNA methylation, which is progressively reestablished as embryogenesis carries on. And again, when you go into the primordial germ cells, you erase most of this demethylation to establish a new program in both male and female gametes. So in mammals at least, we know that DNA methylation, which is a key epigenetic mark, which has potential for memorization over multiple cell divisions, is indeed reset dramatically at least twice during development. And that's how you can get the same program reiterated at every generation. Now what we have in a mouse, for instance, is instances in which this resetting somehow seems to fail. And these are two examples, indeed, in the lab mouse, where you have two genes here, which govern different phenotypes, which have the same DNA sequence. So these are exactly the same alleles. Sorry, this is just one allele of a gene, which is characterized by a repeat sequence inserted into the middle of the gene. And as you know, repeat sequences are very abundant in our genomes. But this sequence now can exist in two states: methylated, here, this black lollipop, or unmethylated. And depending on the methylation state of this repeat sequence, the gene is on or it is off, closed chromatin or open chromatin. And somehow these two epiallelic states can now be transmitted through generations. So they are not reset at every generation, unlike this diagram that I showed you. They fail to reset, they basically stay methylated throughout. And this they do through multiple generations. So here we have now genetics in effect. We have a mutant phenotype versus a wild type phenotype, which is transmitted according to some pseudo-Mendelian laws. Now we start to, of course, depart from truly Mendelian surrogation, but nonetheless, which are caused by a change, a stable change in gene expression, which is not caused by a change in DNA sequence. And we have the same story with another locus called agouti variable yellow. So agouti variable yellow is one allele which can exist in two alternative epigenetic states, and transmission there becomes a bit more complex. It's only through the mother and has some a very high reversion rate. But nonetheless, you can transmit for multiple generations one state without this resetting occurring over the locus. Now, plants perhaps provide the most striking example of such epimutations. That's how we call them, transgenerational epimutations. So here is one example in tomato, where you have, again, just one allele, where you will find this repeat sequence, and that's a recurrent theme in what I'm going to describe today. The repeat sequence, which can be methylated, and here it's methylated in both the wild type and the mutant form of the tomato fruit. So the repeat is constantly methylated, but what you see is that downstream of this repeat, now you have some cytosines which are not methylated in the wild type, and this is a promoter of this particular gene. And here now, somehow, this cytosine has become methylated. Not just one, but a bunch of cytosines. So we believe that there was some spreading of DNA methylation from the repeat into the adjacent sequences. And somehow this spreading, which occurred by accident, we don't know how, then became stably inherited through multiple generations, to the point where now CNR is a true Mendelian trait. segregates, breed through, like any kind of DNA sequence alterations you can have. No single revertant has ever been observed for CNR, so it can be extremely stable over millions of meiosis. No resetting at all here. Yet, when you look at some fruits, what you will find is that often you see small or large revertant sectors, so clearly indicating that in some cells, here it's very clear, early on during fruit development, you had one cell in which now we switch back from this state to this state in one cell. And as the fruit developed, all the progeny of that cell, through somatic cell divisions, kept this unmethylated state. So now you have reverted back stably, but this is somatic reversion. So in a sense, this is the opposite of a program you will have in mammals. In mammals, what you do is you use this memory system through development, and you reset it at every generation. Here, by accident, somehow, you went from this state to this state. You transmit it through generations extremely stably. We have never seen a revertant of CNR, and yet you have no problem in seeing somatic reversion. So the state is stable through meiosis and unstable through mitosis. Quite puzzling. But this is just to illustrate where we can go with this kind of variation. It can be confounded with genetics, with classical genetic inheritance based on DNA sequence variation. And it has yet properties which remind us that epigenetics has this inherent instability in it. But now this is somatic instability as opposed to meiotic instability. Now, so this raises, of course, a huge number of questions, but I think the most important ones are, if you take a given species, a given genome, what is the potential for epimutability across the entire genome? Are all the loci equally mutable? And from what I told you so far, it seems that this may not be the case, because this may very well depend on a very precise interaction between a gene and a nearby repeat. And repeats, of course, move around the genome. They will all not be located in the same place. And so you expect a lot of polymorphism in terms of epimutability potential at a given locus in relation to the exact genomic composition of that locus. The second biggest issue is what determines this very strong transgenerational inheritance in some cases when it is reset in others. Or why do we have, in the case of CNR, this somatic instability versus meiotic stability? These are very unclear. We have absolutely no answer to these questions. And of course, if CNR is just one example, the issue is how much of what we see in nature in terms of heritable phenotypic variation is caused not by DNA sequence changes, but indeed by the kind of changes that we see at CNR associated with an increase or a decrease in DNA methylation over specific sequences. And finally, and this is where the field is a bit muddy and also where a lot of excitement is going on based on very little evidence so far, is what is the role of the environment in inducing this type of variation? Basically, has CNR been induced by a change in the environment, or does it happen sporadically? Can we shift this very stable transmission of CNR through meiosis by challenging the plants with environmental insults? We don't know. So to start to answer some of these questions, and we turn to a system which is ideal in many ways, which is Arabidopsis thaliana. It's a plant species, of course, where you have first a very small genome, which is fully sequenced at least once. So now we have a reference. We can, whenever we want to study epigenetics, we can double check that there is no DNA sequence variation associated with the changes that we are interested in. So we can sequence as often as we need, and we can do that here quite readily because it's becoming extremely cheap to sequence such a small genome. Second, we have many mutants in these plant species which have dramatically altered methylation patterns. So we can start to do, and they are both viable and fertile. So we can do genetics with very, very contrasted methylation patterns across the genome, and then ask all kinds of questions about the stability of these methylation changes and their phenotypic consequences as well. And finally, it's a proper species, so we can go into nature and ask Arabidopsis actually colonized most of the northern hemisphere, so we can ask questions about this type of variation, epigenetic variation, in different contexts. Is it more frequent in harsh climates? Is it less frequent in more temperate climates, etc., etc.? Now, I'm sorry because I realize that here so the audience is extremely diverse, but I want just to put this slide to tell you that we know a lot about the mechanisms by which DNA methylation is both established and maintained in these organisms. So many genetic studies. Now we have an exquisite picture about how you establish methylation over repeat sequences, how you maintain this methylation, and sometimes how you may erase it. And key to all of this system, which is very complex as you can see, which involves what we call RNA interference, so the production of small RNAs which will guide DNA methylation. What you have is this chromatin remodeler, so that's a very important point. We are looking at a modification on the DNA sequence, which is totally dependent on the organization of this DNA sequence into chromatin. So a chromatin remodeler called DDM1, whose function is actually still not very clear, which may help somehow this system to work in what we call closed chromatin. DDM1 orchestrates this machinery. So without this DDM1 function, somehow maintenance methylation over repeat sequences collapses altogether. So we have used this mutant to create a very simple, to set up a very simple genetic experiment. So as I said, we are doing genetics, and so you will recognize immediately here that what we have created, what are called recombinant inbred lines. The thing that we recombine is not DNA sequence because we start with the same accession called Columbia, so they have the same genome, these two parents, but one has now a mutation in DDM1. And because it has this mutation in DDM1, now the repeats across the genome become very, very severely hypomethylated. So we start with two genomes with same sequence, but with extremely contrasted methylation profiles. And then we made an F1, of course, that we backcross to the wild type, just to make sure that we remove the DD1 mutation. So now we are in the F2, in the backcross progeny here. We select all of the individuals, 50% of the progeny, where there is no more the DD1 mutation. So now all the individuals that we selected are homozygous for the wild type DDM1 allele. Okay, so now we are back to a completely wild type genome with no mutation to interfere with DNA methylation. And then the key question is to ask how many of these changes that were induced by DDM1 in terms of DNA methylation, how many of these demethylation changes are inherited and can be passed on through multiple generations to their progeny. And that's basically the test of transgenerational inheritance of an epigenetic defect, of a DNA methylation defect that we induced here at once. And the test is to do it, of course, independently of the inducer. Otherwise, we would never be able to test that. So here we end up with these epireals, so they are epigenetic recombinant inbred lines, and we ask the questions, indeed, how many of these differences in demethylation can we find here? If we find them, do we find them in the proportions expected according to Mendel's law? So basically, we will expect, because we did a backcross at any given locus with differential methylation between the parents, 25% of individuals with the DDM1 hypomethylated state and 75% of the individuals with the wild type methylation state. So we did that, and I'm just summarizing now. Again, I apologize to non-molecular biologists, but it's important to know that all along we understand the mechanisms that go on in this population. We basically found that out of about 3,000 regions that we could very well define in terms of their differences in methylation between the two parents, 1,000 of them at least had inherited the hypomethylated state from the DDM1 mutant parent according to Mendel's laws. So which means that they were induced here, these differentially methylated regions, and now they have been transmitted right to the end of our scheme, 8 generations down this scheme, without any alterations in their transmission. Even though what was used to induce them, the DD1 mutation, was removed in this generation. So now you have true transgenerational epigenetics. So basically, the alteration that you induce can persist. This chromatin alteration can persist independent of what induced it in the first place. And so what we notice is that we can actually explain the difference in behaviors for these different types of repeats. So these DMRs, differentially methylation, correspond to repeat sequences distributed throughout the genome, and why some are inherited in the Mendelian state and others revert back to the wild-type state depends on the efficiency by which they get targeted by these RNA interference pathways that I briefly mentioned before. But what is interesting is that even the sequences that revert have now dynamics of reversions which are quite unlike what you see for DNA sequence reversion. A DNA sequence exists in one state, A, or another state, G, C, or T, and reversion is back to one A, if you are lucky, or to another nucleotide, whatever. But you don't exist in between. You are not between an A and a G. You are A or G. While here you can be in between, fully hypomethylated, DDM1 state, and fully methylated, because it takes quite a while for this wild-type methylation to be reestablished over what we call reverting DMRs. And this reestablishment takes place through sexual reproduction, and it has a lot to do with this machinery that is based on RNA interference, which is most active during sexual reproduction. So first you need to reproduce in order to perhaps reestablish some methylation, and if you do it, you can't do it at once. It takes 3, 4, 5 generations for you to build up enough methylation to now get back to the wild-type level. So even reversion in itself has some very unique properties in this system. But now let's consider what happened in nature. So here it's an experiment that we perform in the lab. We have this DDM1 mutation that we use to induce this potential epigenetic variation, and we establish that yes, there is a large potential, at least 1,000 regions in the genome that can be transmitted according to Mendel's laws. But what happened to DNA methylation variation in nature? There is a lot. And this is now, again, thanks to the possibility of sequencing multiple genomes in Heritability. This is 130 or so accessions taken from all across the globe, for which people have done sequencing not of DNA, but of DNA methylation at single nucleotide resolution. You can do it through a technique called bisulfite whole genome sequencing. And what they found is that genes tend not to be methylated and show very little methylation to start with. This is indicated by this red color. But now when you go for repeat sequences like transposable elements, they are often methylated, dark colors. But as you can see, for any line that you look at, which corresponds to one particular transposable element sequence, you can find evidence of loss of methylation in one or another accession. So there is variation in the natural variation for DNA methylation in nature. Sorry, yeah, I think I said it twice, but that's important to remember that this is now a natural variation. Of course, what you cannot say here is that these variations in methylation states are indeed epigenetic in nature, because they do co-segregate with a lot of DNA sequence polymorphism. We are in natural accessions, so there are plenty of DNA sequence variation as well. So they are always going to be associated with DNA sequence variation. But what we can do now is combine our DMRs that we establish in the lab in the absence of DNA sequence variation with the DMRs in nature and see if there is any overlap at all between these two data sets. And to our satisfaction, what we found is that 40% of what we call stable DMRs in our setup with no DNA sequence variation do have a match in nature, which we can then propose that in nature they do exist in their own right, independent of DNA sequence variation, even though they do co-segregate with sequence variation. The fact that we can establish them in the lab, independent of sequence variation, suggests that they do exist also in that state in the wild. And now, of course, what we find is that if we turn to our DMRs for which we have this very unstable hypomethylated state, so basically a very strong propensity to constantly acquire DNA methylation, you don't expect them to be that often polymorph in nature, because they have a mechanisms to reacquire methylation very rapidly if by any means they lose this methylation by accident. Yeah? And sure enough, these revertant DMRs are the one for which we have the least overlap in nature. So all makes sense. So basically what we have established in the lab reflects what happens in nature. Now, we went one step further. We have genetics that now is based on DNA methylation variation as opposed to DNA sequence variation. So we can derive epi-haplotypes in the same way as we derive genotypes. So here is just one example for one particular epireel and one particular chromosome. We can determine along the chromosome the parental origin of that chromosome based on these epimarkers, because they are stably inherited according to Mendel's laws. And so these define recombination breakpoints, the meiotic crossovers. And so we did that for a large number of epireels and basically used it to derive what we call a classic genetic map. Here you have a genetic map which was entirely built based on epimarkers as opposed to DNA sequence variation. So it's just, it's a bit of an extreme, but it's to illustrate the fact that yes, you can do genetics based on something else than DNA sequence variation. Now we use these genetic maps to go one step further, which is to now ask, is any of this relevant in terms of phenotypic variation? So basically, are any of our stable DMRs associated or causing phenotypic variation, which of course would then be heritable. So we turn to phenotypic analysis using quantitative traits, because we felt that this was the most relevant approach for getting a genome-wide, unbiased view about the potential effects of epimutations. And we did it in different contexts, in a greenhouse, in a lab, in a cross chamber, as well as in an open field. And that's important because you always hear that epigenetics is extremely sensitive to the environment. So here what we wanted to do is to test if we found any heritable phenotypic variation, whether that variation was consistent across environments. And so here is just to illustrate the fact that we had no problem in finding, of course, phenotypic variation, but also in identifying a quite substantial part of this variation being heritable and therefore of genetic origin. Here I'm using the term genetics in what it originally meant, which is simply the study of heritable variation. So yes, we have about 30 to 50% heritability for any of the traits that we looked at in our population of epireels. And now what you can do is go to the next stage, which is simply to try to associate your heritable phenotypic variation with heritable molecular cause. And this will be, in our case, the methylation variation. So we do a classical QTL mapping experiments where we try to associate differences in phenotype with differences in epigenetic states along the genome, among our lines. And again, this is completely genetic, so we basically had, again, no problem in identifying QTLs corresponding to regions of the genome that associate with heritable phenotypic variation, and that explains, in some cases, quite a significant part of this heritable phenotypic variation. And the experiment at the top is important because this is flowering time, which we measure both in a greenhouse and in an open field. And as you can see, the QTLs overlap in the two conditions. So basically, this is the same set of epimutations that is responsible for heritable phenotypic variation, flowering time, in a greenhouse and in the open field. So our epivariants must be robust to environmental influences. Now the challenge is to go from these QTL intervals to the causal DMRs. So to really demonstrate that there is one particular region of the genome with differential methylation that is causing by itself the change in phenotypic values for the traits that we have analyzed. And so there are ways of doing it, which is called fine mapping, and here it's a bit more complicated, but we are now greatly aided by the advent of CRISPR-Cas9 technology, which allows you to target very specific regions of the genome for not only DNA sequence alterations through mutagenesis, but in this case, through epigenome alterations. So we can fuse Cas9 nuclease to a DNA methyltransferase or even a DNA demethylase, and therefore to expect to oppose demethylation or remove demethylation specifically over a single region of the genome, and therefore test whether our DMRs are indeed causing or not the heritable variation. Now, it's all well, but as I told you with CNR, there is this inherent potential for reversion. And we see it in our population with 2,000 DMRs, so basically 2/3 of our DMRs that actually do revert, even though they do it progressively, they do revert. So what we wanted to see is, what about the stable DMRs? Do we have any evidence that, just like the reverting DMRs, they have indeed a potential for reversion, which would be much higher than the potential for reversion you have with the DNA sequence? And yes, when we look carefully at our EP haplotypes, we had no problem in identifying a few DMRs that we define as being stable, segregating in a Mendelian fashion at the population level, but which in a given particular EP reel now are in the wrong state. So basically, this is clearly a DDM1 derived interval, and yet these two stable DMRs exist now in the wild type state. So we do have spontaneous reversion, even for stable DMRs. And we went on to analyze that more. We have about half of all our stable DMRs for which we have evidence for at least 1 or 2 EP reels in which they have reverted to the wild type state. So the rate of reversion is pretty high. We believe that probably most of our stable DMRs in 100 or 200 generations will go back to the wild type state. So if we propagated our EP reel population, which now we can use to do genetics in 200 generations, we're pushing a bit the argument here, but in 200 generations, we'll have no genetic diversity anymore. It'll be back to the wild type parent that we started with, perhaps. So this is to illustrate that. So even these stable DMRs that somehow can persist in the hypomethylated state independent of anything, progressively will require, over now many generations, we don't know how many, will require the fully methylated state. And that makes sense because we started with these DMRs in a fully methylated state in our starting strain, the wild type strain. So they do, and we know that these DMRs do exist in nature, mostly in the wild type strain state. Yeah. So we know that DNA methylation is a default state for repeat sequencing. So what is interesting to consider is the genetics that can happen between these two situations. And of course, as with the rapidly reverting DMRs, we expect progressivity as well. So we will have all kind of intermediate methylation states which may have intermediate phenotypic effects between fully methylated, unmethylated, and fully methylated. So that complicates our picture of genetics, but this is perhaps closer to the real life. So I think it's time that I conclude, probably, yeah. So forget about the environment because there was nothing going on. And these are the take-home messages. So plant genomes, perhaps more than mammalian genomes, have a huge potential for epimutability, we believe, especially because many plant genomes are huge in terms of contents of repeat sequences. You know that the maize genome is mostly made up of repeats, so the potential for generating this kind of epigenetic variation is probably immense compared to other systems. We could show that immutable loci do contribute to heritable phenotype. Of course, we have studied the complex trait, and I think this may be most relevant in natural settings because you are not in nature studying CNR, which is a monster that will probably not survive for very long, so it has no evolutionary impact whatsoever, whereas this type of variation clearly fuels evolution. Now, I didn't mention that to you, but people got very excited based on the results obtained with the natural accessions and natural demethylation polymorphism. They found some methylation in genes itself, not just in the repeat, but in the gene. And they said, oh, well, this is great, even the genes can now acquire or lose demethylation in an epigenetic fashion. Well, we analyzed the data, and we found that there, what they have is DNA sequence variation. A transposon landed in a gene in one particular accession, methylation spread to the gene sequence. This is based on Illumina short-read sequencing. You never map the transposon, you simply map the flanking sequences, which are the gene sequences, and now you call it methylated, of course, compared to unmethylated anywhere else. And you say this is an epigenetic variant. No, it's not. It's a DNA sequence variant with methylation spreading to it. And finally, yes, we have absolutely no evidence whatsoever in our experimental system that the environment does anything. We are challenging our population, we're putting it under very harsh conditions, and yet we haven't seen any evidence of the environment doing much to the methylomes of these different epiroids. And with this, I will leave that for the discussion. I want to thank the people in the lab, and as you can realize also it's a huge collaboration with quantitative geneticists, with sequencing people, etc., etc. And yes, I want to finish with this, which highlights some points that will be useful for the discussion, maybe not now, but this afternoon as well, and which has more to do with what happens in humans. Thank you.